folks, welcome into the ITLS Academy. Myself Dr. Himani, and today we are going to discuss about the yeast. So let's start with the introduction of the yeast. In 1916, Dutch naturalist Anton von Leeuwenhoek first microscopically observed yeast. Researchers were doubtful whether yeast were algae or fungi, but in 1937, Theodore Swin recognized them as fungi. So, in 1957, Louis Pasteur provided in the paper Memorise sur la fermentation alcoolique that alcoholic fermentation was conducted by living yeast and not by a chemical catalyst. The late 18th century two yeast strains used in brewing had been identified: Saccharomyces cerevisiae or Saccharomyces carciverginius. It's like a bottom fermenting yeast. In 1995, a mention was developed to remove the liquid so the yeast could be prepared as a solid block. Let's move on. Next slide. What is yeast actually? So, yeast are eukaryotic single-celled microorganisms classified as member of fungus kingdom. You can see in this picture the structure of the yeast. So this is the microscopically image of mature yeast at the high magnifications. At least 1,500 species are currently recognized. They are estimated to constitute 1% of all described fungal species. Yeast are unicellular organisms that evolved from multicellular ancestors, with some species having the ability to develop multicellular characteristics by forming strings of connected budding cells known as pseudophyte or false hive. Each size vary greatly depending on species and environment, typically measuring 3 to 4 micrometer in diameter, although some yeast can grow to 40 micrometer in size. Most yeast reproduce asexually by mitosis. This is very important things, and many do so by the asymmetrically division process known as budding. The budding yeast is called true yeast, are classified in the order Saccharomycetes with the phylum Ecomycota. With their single cell growth habit, yeast can be constructed with the molds which grow hives. Fungal species that can take both forms depending on temperature or other conditions are called dimorphic fungi. So next move on, the next slide is like nutrients and growth. But the yeast species Saccharomyces cerevisiae convert carbohydrate to carbon dioxide and alcohol in a process known as fermentation. Saccharomyces cerevisiae is also an important model organism in modern cell biology research and is one of the most thoroughly studied eukaryotic microorganisms. Other species of yeast such as Candida albicans are opportunistic pathogens and can cause infection in humans. So about the nutrition and growth, yeast are chemogenotophers as they use organic compound as a source of energy and do not require sunlight to grow. Carbon is obtained mostly from hexose sugar such as glucose and fructose, or disaccharides such as sucrose and maltose. Some species can metabolize pentose sugars such as ribose, alcohols, and organic acid. Yeast species either require oxygen for aerobic cellular respiration, it called obligate aerobes, or are anaerobic but also have aerobic methods of energy production. This called facultative anaerobes. Unlike bacteria, no known yeast species grow only anaerobically. Most yeasts grow best in a natural or slightly acidic pH environment. Yeast vary in regards to temperature range in which they grow best. For example, Leucosporidium frigidum growth at -2 to 20 degrees centigrade, or it's called 28 to 68 Fahrenheit degree, and Saccharomyces telluricis at 5 to 30 degrees centigrade, or it could be the in the 41 to 95, and Candela slope at 28 to 45 degrees centigrade, it could be the 80 to 113 Fahrenheit. And now let's move on the ecology. What is the ecology of the yeast? Yeast are very common in environment and are often isolated from sugar rich material. Example include occurring yeast on the skin of fruits and berries such as grapes, apple or peaches. Some yeast are found in association with soil and insects. The ecological functions of biodiversity of yeast are relatively unknown compared to those of other microorganisms. Yeast include Candida, Candida albicans, Rhodotorula rubra, tor, Torupasis, and Triscosporum cutaneum have been found living in between pupil toes as a part of their skin flora. Yeast colonizing nectaries of the sticking halobri have been found to raise the temperature of the flower, which may aid in attracting pollinators by increasing the evaporation of volatile organic compound. The yeast has a negative effect on a bacteria that normally produce antibiotics to kill the parasite. So many affects the ant's health by allowing the parasite to spread. Certain strains of some species of yeast produce protein called yeast killer toxin that allow them to eliminate competing strains. This can cause problem for the winemaking, but could potentially also be used to advantage by using killer toxin producing strains to make the wine. Yeast killer toxin may also have medical application in treating yeast infection. So now we will discuss about the reproduction in the yeast cell. Yeast generally reproduce by asexual methods such as budding or fission. Yeast lacks sex organs, and sexual reproduction in yeast is highly variable. Two type of Reproduction occur in this yeast cell. First one is called budding, and the second one is called sexual reproduction. So let's just discuss about the budding. What is budding reproduction? So it occurred during abundant supply of nutrients. You can see in this picture. There is the in this picture you can see this is the parent yeast cell, and this parent yeast cell convert into the bud forming. You can see here this is the start of bud forming, and is divided into two parts. This is the buds remain attached here, you can see, and bud nipped off. This is, you can see here, other buds formation is occurring here, and here this part is bud ripped off separately, and this is the new yeast cell. So it occurs during abundant supply of nutrient, budding reproduction, and parent nucleus divides and move toward daughter cells. This one, parent nucleus cells divide and move to the daughter cells. Enzymatically activities increase during this phase. Increased Turner pressure act on weaker part of cell wall, and bud erupts. You can see here. Increased Turner pressure act on weaker part. This one you can see here, and the weaker part of cell wall, and bud erupts, and the formation of new yeast cell is there. Let's move on the next slide. Second type of reproduction is sexual reproduction. Sexual reproduction is highly variable in yeast. Three different patterns of life cycle found in different genes. First one is haplodiplobiontic life cycle. Second one is the haplobiontic life cycle. Third one is diplobiontic life cycle. So let's start the discussion about the haplodiplobiontic life cycle. What is this actually? So you can see in this picture, this is the karyogamy. First start this one. This karyogamy is divided into two acts, and then is converted into the somatic cells. Then after meiosis is converted into the axis, and this axis is converted into the ascospherospheres, and this again divided into the somatic cell, and the somatic cells convert into the copulation, and these copulation form the plasmogamy. So in this cycle occur in Saccharomyces cerevisiae. In this life cycle, somatic cells of yeast exist in two forms: haploid dwarf cell and diploid large cell. Haploid cell contain two mating types, or mating type A and mating type alpha. So during favorable condition, each of the haploid cells multiply by budding only. You can see in this picture. So multiply by the budding only. If these mating types comes in contact with each other, they form gametangia and start sexual reproduction. Fusion of these two haploid cells form large fusion cells called plasmogamy. Nucleus also fused by karyogamy. You can see in this picture. Nucleus also formed by karyogamy to form zygote. Zygote multiply by budding and form several diploid cells. You can see here. Several diploid cells are present here, and these diploid cells are larger than haploid cells. Like haploid cells, these large diploid cells also live independent life and reproduce by budding. Under unfavorable condition, diploid large cells become spherical and directly behave as ascus. You can see in there. Again, I am repeating. Under unfavorable condition, diploid large cells. These diploid large cells. These diploid large cells become spherical and directly behave as a ascus mother cells, and nucleus of ascus mother cells divide by meiosis to form four haploid nucleus. You can see here. These are the four haploid nuclei. Out of four nuclei, two belongs to mating type A. These two belongs to mating type A, and these two belongs to mating type alpha. So each nuclei gather some cytoplasm and become ascospore. These are have some cytoplasm, and this gathering some cytoplasm and become ascospores. These are the ascospores. Each ascospore is globular and thick walled structure. You can see in this picture. Thick walled structure is present here, and released by rapture of Ascal's wall. This Ascal's wall is ruptured, and then after rupturing this Ascus wall, ascospore germination to give haploid drop cells. So next, move on the second type of second types of sexual reproduction is haplobiontic life cycle. In this picture, you can see here, this is the ascospores, and this ascospores convert into the different type of cells, and you can see here these convert into the haplophase, and this haplophase again convert into the diplophase, is converted into the X, and this convert into the 2X form, and after meiosis process, you can see here the ascospores and ascus are present here. So this type of life cycles occur in the cyclosporum psychospormosia in haplobiotic cycle. Somatic haploid cell multiply by fission, producing number of haploid cell. During sexual reproduction, two sister haploid cells behave as a gametangia and produce weak-like structure. You can see here. This is the weak-like structure, and this the weak of the two gametangia fused and wall at the point of contact dissolved to form canal called conjugation tubes. This called is conjugation tubes. You can see here. Nucleus of two cells move and fuse to form diploid zygote. You can see here, this is the diploid zygote here. And the zygote directly functions as an ascus mother cell. You can see here. This is a meiosis process, and nucleus of ascus mother cells divided by meiosis. You can see in this picture, Nucleus of ascus mother cell divide by meiosis forming four haploid nucleus. These are the four haploid nucleus. Nucleus are ascus mother cells and divided by meiosis forming four haploid nuclei, which themselves divided by mitosis to form eight haploid cells. You can see here these are the four nucleus of ascus mother cells. These are the four haploid nuclei, and these are the other four haploid nuclei. So by mitosis to form eight haploid nuclei here, each eight nuclei gather some cytoplasm is present here and become ascospore. You can see here become ascospore, and this ascospore released by rupture of ascus, and ascospores germinate to give haploid cells and continue life cycle. Next, move on third type of cycle. This is the diplobiotic life cycle. In this picture, you can see here spotus mycelium. This convert into yeast cell, and yeast cell by the meiosis process is converted into the haplophases, and this is the ascus with ascospore. You can see here, and by the fusion between ascospores plasmogamy, you can see here this is the plasmogamy, and then this converted into the diploid cells formation. This called carbogamy, and this carbogamy converted into the diploid cells germination, and this diploid cell germination is converted into the spores mycelium. So the life cycle is found in Saccharomyces ludwigi, and in diplomatic life cycle, diploid somatic cells multiply by budding and fission. Under certain conditions, these somatic cells directly function as ascus. You can see here. Under certain conditions, these somatic cells directly functions as ascus mother cells, and nucleus of ascus mother cells divided by meiosis. You can see here these are divided by the meiosis process, and to form four haploid nuclei. Here is the four haploid nuclei. These haploid nuclei gather cytoplasm and transform into ascospore, four ascospore. You can see here. Four ascospore is here. So out of four ascospore, two are mating type A1 and two are mating type of A2. These two mating ascospores fuse within ascus to form diploid zygote. You can see here. So the diploid zygote germinates with ascus producing a germ tube. These germ tubes break the ascus wall and function as a diploid spores mycelium. This spores mycelium on budding gives gives a um, sprout diploid cell. You can see here. These are the diploid cell germination, and this give rise the sprout diploid cells, which give diploid somatic cells. So this was the discussion about the asexual type of cycles in yeast cell. So now question is that how yeast work. Yeast need three things to thrive: moisture, food. Food could be sugar and warm temperature. When yeast is hydrated and give sometimes to feed on it being growing, yeast favorite food source in sugar in its various forms. Yeast does not just feed on white sugar, but it can feed on carbohydrate in any form. When yeast is hydrated and gives some food, it being metabolizing the sugar producing carbon dioxide and alcohol. In baking, the carbon dioxide bubbles are trapped within the structure of the baked good, causing it to rise. Alcohol, the other byproduct of yeast fermentation, is also produced during the bread making process, but evaporate the bread bakes. When making alcoholic beverages, yeast is allowed to ferment for a much longer period of time, causing it to produce more alcohol. Yeast also thrive in warm temperature. It could be 95 Fahrenheit or 95 degree centigrade. Oh, sorry, 35 degree centigrade is the temperature at which yeast most actively grow. Temperature cooler than this will cause the yeast growth to slow down and yeast cells will start to die at 135 Fahrenheit or 57 degree centigrade. Sorry, this is the centigrade. 50 degree centigrade temperature at the higher. So now we will discuss about the applications of the yeast. First application of the yeast in preparing beer. Yeast is added to malted grains and encouraged to ferment in order to produce alcohol. The type of yeast used will affect the type and flavor of beer produced. Saccharomyces cerevisiae, also known as top fermenting or top cropping, is fermentation at a higher temperature and produce sweet or fruity beer. Bottom croppers such as Saccharomyces pastorans fermented at lower temperature and are used to make larger. Next application is preparing wine. So, yeast is naturally present on the skin of grapes and can be used to naturally ferment grape juice into wine. Despite the naturally present yeast, most wine today have a pure culture, usually Saccharomyces cerevisiae, added to them to produce a more consistent and controllable result. There are many different strains of Saccharomyces cerevisiae yeast, and each will produce a unique flavor characteristic in a finished wine. Next application is preparing in non-alcoholic drinks. Root beer and other soft drinks use yeast to add flavor, but the fermentation process is stopped before the drink because alcoholic. This means the drink are much sweeter than their alcoholic counterparts and contain more carbon dioxide. They also normally have a very low alcohol content, although this is usually around 0.1%. Next application in preparing bread. The most common use of yeast has been in the making of bread. The yeast react with oxygen and help leave bread or make it rise. During Passover's, Joel's pupil will remove the yeast from bread to make flat bread. There is evidence the ancient Egypt, Egyptians used yeast to make bread around 4,000 years ago. Next application is as a nutrition. Raw yeast is not recommended for consumption in any large quantity as it can proliferate within your body and have negative effect. Once pasteurized yeast offer a host of nutritional benefit, Yeast is a good source of protein, B vitamins, and minerals. Yeast, intended as a nutritional supplement, is often sold as nutritional yeast or yeast extract and can be used as a flavoring supplement or other way. Yeast extracts such as marmite or vegemite have a cult, cult following due to their unique flavor and variety of uses. It could be useful in scientific research. So, due to the cellular makeup of yeast, many scientists use it to learn more about human genetics. Study of yeast culture lead directly to the mapping of the human genome. It could be beneficial in biofuels. Most recent yeast has been used in the production of biofuels. This is because the yeast turns sugar into ethanol, which can be used as a diesel substitute in vehicle. This process it goes through is identical to that of making beer or wine. And as a skin moisturizer, it could be also very beneficial. It can work beautifully together with collagen, a major protein in skin that is responsible for its firmness. The proteins in yeast can help fight skin lacty and wrinkles, as yeast is already in several type of skin moisturizer. Next application is as a skincare treatment. Yeast can contain some of the minerals, amino acid, and vitamins that your skin may be missing. Use it as a skin treatment by adding some baker's yeast to your skin cream. So, guys, hopefully you all enjoy this lecture. If you have any queries, then you can just ask me, and you can just follow us by the Instagram, Facebook, Twitter, YouTube, WhatsApp, or by the LinkedIn. So, guys, thank you very much for listening me. Thanks a lot.